Hi, everybody. Welcome to Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from New York, Amy Rosenfeld.
Hello, Vincent. How are you today?
Okay, and I'm not going to ask how you are, I think. Best not to.
Yeah, best not to.
So I heard that the governor's race in New Jersey was finally decided, right?
Yes, first time a Democrat has won a second term in like 40 years since 1977. What is wrong with your state? Like, why did it take that long? Like, there's no reason that this should have ever been a discussion. I mean,
No, it should never have been this close. And the thing is, folks who are here tonight, welcome everybody. This race was close because the other party candidate ran on a platform of no vaccines and no masks.
no, he ran on a platform of no taxes.
And no taxes.
If you don't, so he ran on an ad that said that your governor apparently said, if you don't like taxes, New Jersey is not for you. So he ran on the he ran on a platform that your state, your property tax is absorbent and is not cohesive. Cohesive isn't the right word. It's not compatible with modern suburban life.
I tell you, my taxes are very high. Are yours?
I live in New York City. I don't pay any taxes because I don't own anything. Well, I do pay taxes, but I don't pay property tax. I don't own anything.
All right, let's move on to Q&A. Did you hear about this deer study in Iowa, by the way?
Yes, from Penn State, yes.
And I think it was Iowa. Yeah, Iowa. And 90% of the deer are positive. Wow. That's a lot of deer, right?
Seems to me to be a lot of deer, especially when you're in hunting season.
And Philip says, cook the deer well done. The problem is when you hunt it, you're going to get, you have potentially could get infected when you hunt it. So,
well, when you bleed it.
Yeah. Do you have to bleed it out before you bring it home?
No, usually you just drop it to the roof of your car and then you bleed it in your garage, or at least that's how Ira used to do it.
So you've had some familiarity with this process,
yes.
I take it.
Yes. Yes.
Amy's done everything. Amy has done everything.
I didn't do it. My father did it. My father went deer hunting. And then if he shot a deer, depended on what the agreement was with the people. Mm-hmm. Sometimes they cut it up before they left. But sometimes he brought it home attached to his roof of his car, and then he hung it from the ceiling of the garage to bleed it out.
Aye.
Wendy was not pleased.
Did you stay clear of that?
I did, but Wendy did not. Wendy walked into the garage one year when it was bleeded out and was not happy.
Steve wants to know, does a 90% efficacy mean 9 out of 10 people will never get infected, or that a person doesn't get infected in 9 out of 10 encounters?
Means none of that.
What does it mean?
As far as I can tell, it means that it only has to do with severe disease. The development of severe disease has nothing to do with people getting infected or encountering the virus.
Right. So 90% does refer to severe disease, hospitalization, death, and it's based on looking at people who are in those conditions and seeing how many are vaccinated and how many are not.
It has nothing to do with getting infected or encountering the virus.
And it may not apply to you. It may be done in somewhere else where things are different.
Huh?
Well, if it's in Arizona, and the healthcare system is really good,
of the vaccine change, it can't change.
Even if your healthcare system is not good.
No, it can't change.
All right. When a virus is being attenuated to make a vaccine, how are the desired variants identified and sorted from the swarm, and how are they tested for desirable properties?
Means
good
it's
question.
targeted. The fact is, it's not a targeted thing. It's empirical. Sabin was completely empirical. He rapidly passaged virus through the kidneys.
Yeah, then he checked. He would plaque purify and check plaques in monkeys to see if they had lost some virulence, and then go back and do it again. Now, today, in theory, you should be able to introduce specific mutations, and there's one vaccine I'm aware of. It's a dengue vaccine. It's in development, where they deleted 30 bases from the 3' non-coding region, and the virus can still reproduce, but it's attenuated. And so that's the sort of thing you can do in the modern era. You don't have to pass hundreds of times.
But that's okay.
What do you mean you can't do it in the modern era?
So in order to do that, you had to spend 50 years understanding dengue biology.
That's right.
So it's not like you're going to go out and get a novel flavivirus and say, okay, if we delete 30 bases of the 3' untranslated region, it's going to attenuate this virus. If it were that easy, then we should all be making C472 similar mutations in all coronaviruses and call it a day.
By the way, the 30 base deletion in the 3' UTR, we talked about that today in class. That prevents the formation of the subgenomic flaviviral RNA, which is needed for pathogenesis. So you remove that, you can't make that small RNA, which is made by the exonuclease XRN1, and the virus is attenuated. Amy, I wonder what you think of Eric Lander's work. Shouldn't he have done something public by now? He seems obscure to me. Anything but obscure, right, Amy? He's a science advisor to the White House, correct?
Yes.
Or something like that. Maybe he's got some other title.
He's the science advisor to the White House. He sits. It's part of a cabinet. He's a part of the cabinet now. Yeah, I don't know what you mean by he should have done something by public now. What do you want him to do?
Yeah, usually the science advisor, and he's the president of he's the head of OSTP, Office of Science and what's the T? Technology Development. Usually they're quite quiet. They just run events, and you don't really hear from them.
Well, what do you want them to do?
I don't want him to do anything for myself. But, as he is a rather prominent American scientist, you would assume you would hear from him. But I agree. I don't think he needs to do anything that's visible. He's doing things behind the scenes.
He's sequenced. That's what he's known for.
He's a genomic person, yeah.
I don't get it.
How purely lipid are lipid droplets in the cell? Can they take up lipophilic drugs?
They're pure lipid droplets. Yes, they can take up anything that likes to be encased in fat.
Don't they have a protein, proteolipin?
Sometimes they do, but other droplets are
pure lipid, yeah.
Yeah, fatty acid droplets that are that like Raoul and Orley characterized are just lipids. They're just conglomerates of lipids.
Can they take up lipophilic drugs? I would guess so, right, Amy?
Yeah.
Elena wants to know, how long does it take to write a grant proposal?
Seriously?
About a month, right?
Seriously?
Longer?
Yeah, way longer.
Maybe
You're lucky if you can do it in a month. First, you have to think about what it's about. Then you have to figure out whether or not you have any data to support your hypothesis, right? First, and then you make a hypothesis. Then you have to figure out if you have any data to support it, right?
Exactly.
Then you have to figure out. So then you have to imply the cheater philosophy. It's true that you have to sit there and think about all the ways that the reviewer is going to poke holes in your data. And then you go back and try and do those experiments before before the deadline of the grant, right?
Right. Yeah, for
And
sure.
still, you have not figured out the cheater philosophy,
And you
right?
think, here's the thing, you think you've written a nice proposal, you go over it many times, more than one person, and then someone else reads it, a few other people read it, and they find things that you never thought of, which are not necessarily right, but they
Right,
find
but
them.
that's the cheater policy. That's what
Yeah,
I'm
right.
talking about,
Exactly.
right? It's the cheater philosophy.
Um,
So, how long does it take to write a grant? When I finish a grant, I'll let you know.
well, you finished many.
They're not finished. Finished is when they get funded and you have a paper and a variety of other things.
Well,
Very few.
you've had a number of those.
Yeah, but it took years.
Can subvariants have significantly different properties affecting infectivity, transmission, and virulence? If so, could this, say, accelerate the right side of the bell-shaped curve?
What's on the right side of the bell-shaped curve?
I don't know. That's when the infections go down, right?
Yeah, I guess.
But they could have changes in these properties. But to prove it is difficult. It's not something that you can assess by looking at an outbreak and saying, oh, yes, a lot of people got infected. There must be differences in these properties. No. And I have to remind everyone that, early in the Delta outbreak, numerous preprints came out saying the virus was associated, a variant was associated with more severe illness. And now the CDC has come out last week and said, no, actually, we don't think it is. So this is hard stuff to sort out.
Really?
Thank you. Isn't it shocking? Thank you, Miriam.
I'm still flabbergasted that it's difficult to sort out. I mean,
We have to do experiments.
really?
And that's the yes, that's the problem. Epidemiologists make observations. That's their job. But these kinds of claims require experiments. Thank you, Miriam, for your contribution and support of the incubator. Someone wants to know if we can have Paul Offit on a Q&A. So here's the thing, folks. I was actually surprised that not many people came to Daniel's Q&A. We get a lot here, 600 some, and that's what Daniel got. And that's great. But I just had this feeling, there's so many people that have questions. Why didn't they come? So I don't know about Offit.
Well, I mean, let's get real here. There's very little publicity. You don't market it at all.
I don't market it at all. You're absolutely right. There's zero publicity.
Right.
Yeah.
So if you want people to come, you have to market it.
Yeah, you're right. You're absolutely right.
And so that's one thing.
Okay.
And Offit, I mean, I like Offit. Some of what he says, a lot of what he says, I agree with. But he was just on Twitter, like less than 3 months ago. Is he going to say something different?
No, he was on Twitter before the booster story came up, so he could.
No, I think you had him for the booster story. I think you discussed it, which reminds me, I don't understand some of the stuff that you talked about on Twitter on Friday. I don't understand why you and Rich would say that the vaccine is better than being after infection with the virus in the broadness of the response, because that's not what has been shown. I don't know why you guys didn't criticize the paper for that statement, because that's
Is that
technically
what they said?
Yeah, and that's not what Paul and Theodora have seen. That's not what other people have seen. So I mean, and then you entertained Rich when he said, oh yeah, this just goes to prove how much better vaccination is than infection. And it was like, where have you been? Like, that's wrong.
Well, you don't get as sick from vaccination, do you?
It's not about sick. It's about whether or not the breadth of the antibodies in the repertoire are as broad.
Okay. Well, I didn't think I said that, but I stand corrected if I did. Thank you, Sandy, for your contribution. Very much appreciate it. Thank you, Vanity, for the picture. You sent a picture of a cartoon with a couple scientists sitting at a bench in a lab, and someone said, we've been working at this for a number of years, but these people on Facebook seem to have some insight. Yeah, that's pretty funny. I've seen that before. I've seen that before. Many people are happy that you're here now.
Good.
At what point would you two... Sorry, go ahead. At what point would you two personally consider a third dose booster? When would you consider a booster, Amy?
When you can demonstrate that the immunity from my first 2 boosts now doesn't protect me at all, that I have a greater than 50% chance of getting infected and dying. And I'm not clear how you're going to do that.
So you have to look at the data that's arising.
Well, I'm not clear how you're going to do that. Because once you induce, okay, are we boring you?
No, I was looking at a paper on this very subject. I'm not bored.
Well, considering the fact that the virus is going to become endemic and continuously circulating, which means you have a continuous probability of being exposed, I'm not really clear how you would prevent stimulation of my memory response.
I would consider a third dose if I see the effectiveness against severe disease and death and hospitalization drop below 90% for the Mrna vaccine, which is what I got. And I haven't seen that yet. What do you think of the idea that the MMR, measles, mumps, rubella vaccine offers some protection against COVID?
Wasn't that all the innate immune response stuff, like of Chickenpox, like a year and a half ago?
Yeah, before we had COVID vaccines, there were these observations that a number of attenuated vaccines, MMR, oral poliovirus vaccine, could confer some temporary protection, maybe a few months, and the idea was it was thought to be a general boosting of innate immunity. But I don't think anyone officially tested that in a trial of any sort. I think it's plausible. And Chickenpox's mother in the Soviet Union, the former Soviet Union, actually did experiments or trials to test oral polio vaccine against a variety of illnesses, and the data looked reasonable. So speaking of endemic, some at the London School of Tropical Hygiene and Tropical Medicine say the UK is nearing endemic equilibrium and cases will peak in November, even with no restrictions, no masks or vaccine certificates, and winter on its way. Do you agree? What do you think, Amy?
Peak? I'm sure that there will... isn't there an outbreak already in England? In the UK?
There's currently an outbreak, yeah. Well, there's cases increasing, right?
Right. So I would say that you see, this is November, you see increases of cases. There are no restrictions. There's no masks. I don't know what vaccine certificates is going to do, but okay, we'll give them vaccine certificates. So I don't know how you're going to decide if you're near endemic equilibrium, because isn't that where you can you see very few cases? Isn't there something about some significant number of cases per something or other? I really don't understand what the definition of endemic means.
Well, endemic simply means you're not in the outbreak mode continuously like we are now. You have the virus is always present, and there are periodic outbreaks, epidemics now and then, but not a global situation here. But the virus is there. That's what endemic means. It's continuously present.
How do you know that? Well, I don't know that I would use it as it doesn't spike into a pandemic, but okay, that's fine.
I don't know about a pandemic, but an epidemic.
Well, that's what you just said.
No, we don't have. That. Pandemic is not compatible with endemicity. So influenza is endemic. In the U.S., for example, the virus is always present, and in the winter we have outbreaks, right?
Yeah.
That's endemicity. And so whether SARS-CoV-2 is going to do this? Who knows? I think predictions are very difficult to make of these sorts. Eventually, it will become endemic. I'm not sure now is the time. They're at very high percent vaccination there, but there's still a lot of susceptible individuals, so there may still be explosive periods. I think it's very hard to know.
Explosive periods is what, and you just finished telling me an endemic can accommodate. We have explosive periods of flu every winter, but it's quietly circulating throughout the year. But we have explosive epidemics.
Those are endemicities. Yeah. So you're agreeing with the London School of Hygiene?
Well, I'm not. I don't know that we're anywhere near achieving endemic equilibrium. I'm just saying it could be that it could be that you could, but I have no idea.
Okay. Fair enough. Barry says, I get what an antibody titer is, but what does a correlate of protection look like for B or T cells? Number of B or T cells per kilogram of body weight or mil? How do you measure it? Flow cytometry?
Well, I think the extrapolation is one antibody is a representative of one T cell. One B cell. Because a B cell makes only one antibody that recognizes one epitope. So it would be a direct correlation. If you have an antibody titer of 100, you have 100 different B cells.
So the problem is that for antibodies, it's easy to do a neutralization assay. For measuring B or T cells, you actually have to have a peptide, and you ask if it binds to the B or T cell. You need to know if they're virus-specific B or T cells, right? So the assay is not as easy to do. And actually, that's a good question. What the measure would be is not clear, because there is a measure for antibodies, but we don't have one.
Well, as I just said, one antibody is per B cell, right?
Yeah, so you would just
it's a
look
direct
at specificity.
Correlation.
Yeah,
right.
Yeah, but what about T cells?
T cells are much harder, right?
Yeah. T cells, you have a T cell receptor that will recognize a peptide, and if it's a viral peptide, then that would be a virus-specific T cell.
Yeah, but you have to harvest them, you have to stimulate them, you have to do interferon gamma.
Yes.
It's the whole thing about why it's hard to do, why antibodies were the focus of this pandemic, is it was too difficult.
As Florian Kramer said in an article, he said, it's much easier to do neutralization assays, so they're clinically useful. It's very hard to do T cell assays, so they're less clinically useful. It's not even clear what the standard would be.
It's not even clear what the standard for the neutralization is.
That's right.
That's fine.
If memory B cells make antibody, do their kinetics vary person to person? Is it age-related? Does an older person make flawed antibody from old B cells? Fewer cells make antibody slower? So yes, their kinetics vary, and they're age-related. You can lose memory B cells with age, and it depends on the vaccine. So some vaccines induce short-lived memory B cells, other longer.
It doesn't depend on the vaccine. It depends also on the pathogen, because there are a lot of pathogens that we make memory B cells against that you don't have any vaccine against.
It's also dependent on the adjuvant or not. One study in Science by Ahmed showed that unadjuvanted influenza virus, at least in mice, didn't induce memory B cells, whereas
I think he did it in people. I think
it was mice.
Well, I think he correlated it with
people
because I believe he bone marrow tapped those people.
Thank you, Justin, for your contribution. How long does a fully mRNA vax person have to quarantine from an unvaxxed child if exposed? Singapore study says risk is short. Should distancing 3 days be more than enough? I would err on the longer side. I'd go 5 to 7 days. What do you think, Amy?
Depends on who that 3-year-old is. If that 3-year-old is my kid or some neighbor's kid.
Why would it matter?
Well, I'm going to take care of my 3-year-old child, right?
Okay, so what would it be for your kid?
I don't know if I would quarantine or if I would wear a mask. I might just wear a mask if it's my kid and I'm taking care of it. If it's the neighbor's kid, I don't need to see it every day, so then I would quarantine.
Yeah, I agree. I think a mask is a good idea. Yeah.
I think it just depends on what this 3-year-old child is.
Yeah. All right. The Lancet, October 29th mRNA booster study. Did you look at this, Amy?
No.
So I just brought it up here. Effectiveness of a third dose of Pfizer COVID vaccine for preventing severe outcomes in Israel, an observational study.
Oh, another Israeli study.
So you have to be careful with these observational studies. They basically looked at people with a third dose and looked at hospitalization and severe disease, and they say that it was effective at protecting individuals against severe COVID compared with receiving 2 doses 5 months ago.
And how long after the booster?
Let's see. Vaccine effectiveness evaluated at least 7 days after receipt of the third dose.
So you're comparing people who got a third dose 7 later with people who have only 2 doses 5 months later.
5 months ago.
Okay.
Yeah.
That's good science.
And here the effectiveness is against severe disease was 93% for the third dose, compared to 81%.
Well, then you
5
just
months ago.
then you just said if it dropped down beyond 90, you'd get a booster. So this just tells you to get a booster.
Well, I don't believe that these data, frankly. I think
Well, it's not a fair comparison.
right.
Now, if I looked at 7 days after the second dose and I saw it was 93%, and 7 days after the third dose it was again 93%. I don't know what I'm supposed to do with that information.
Oh, what is it 5 months after the third dose?
Right.
Maybe it's 81% again.
Exactly.
In which case you've done nothing with the booster.
Right, exactly. That's why I'm saying it's not the right comparison.
Yeah, okay. There you go. Amy, M-Pro cleaving Nemo resulting in endothelial cell death. What are the implications? M is a SARS-CoV-2 protease, Amy?
Yeah. Okay. I guess you have vascular problems.
Are you convinced that there's SARS-CoV-2 reproduction in endothelial cells?
No.
I've never seen good evidence for it.
I'm not convinced of anything.
And
but that wasn't the question.
This is probably endothelial cells in culture, right? And so my point is,
yes,
that doesn't tell you if it's happening in vivo.
Yeah,
so
I don't know.
implications are limited.
I don't know what this means.
After furin cleavage, S1 is still weakly bound to S2. Is anyone developing a pill that completes the separation so it can't pull the virus along with it when it binds ACE2? That sounds like a tough target, frankly. And I don't know anyone who is looking at that kind of inhibitor. Do you, Amy?
No.
You've mentioned before that the most prestigious journals are not necessarily the most reliable in terms of solid research. What other journals would you recommend that are better? Well, this criticism holds across all journals, right? Everything a journal publishes is not necessarily solid research. And I think you can have other, in virology, there are other journals, and they can be problematic too. So I wouldn't say they're better. I think they all publish, and you have to have a critical eye about evaluating the work, right, Amy?
Yes. Oh, I like this random dude. Amy can't wait for it to be 8:45. Well, I'm glad that random dude knows what I'm thinking.
Amy is here, dude. She likes to be here with us.
I'm glad to if he could figure out what I'm thinking now, he gets a gold star.
Okay.
Seriously, it's kind of tiresome that you say that people have the audacity to sit here and tell me what I'm thinking. If that's true, then we should have random dude answer all the questions.
All right, I took random dude away. Nobody insults Amy.
Murphy's team says Bay stayed home and the pissed off mass people came out in droves. Sure.
It may be, right?
Sure. I really think from all the research, it really had to do with money and the fact that your property taxes are very high and nobody wants to keep paying such high property taxes.
If the first two Pfizer shots are now FDA approved, why is the booster an EUA?
Because it's an unknown quantity, right?
Yeah. I mean, it's still under evaluation, right?
Right.
I had my Pfizer booster. Do I need to wait 2 weeks again for the antibodies to peak, or will it be sooner or later? What do you think?
No, it's going to take it'll take still some time.
Yeah. And you do pay property tax through your rent. Yes, in New York City, that's the way it works. Rent or your monthly carrying charges if you're in a co-op. Yeah.
Right.
Pfizer has been under fire on social media lately. Whistleblower says that vaccine data is falsified. Allegedly, they added non-clotting agent to the children's dose. Thoughts?
I don't know
Who knows?
anything about it.
I mean, this is an allegation that has to be investigated. The FDA will investigate, right? We'll find out what happens. I don't have any insight, but I do know that the vaccine's working, right? And it's quite safe. So you have to balance that over the allegations, but we'll see. There are several studies on N protein causing inflammation in ARDS. Are you aware of S causing inflammation? Aware of any of that, Amy?
No.
No, I'm not either. But you have to be careful about isolated proteins doing things, right? I mean, they might do it in the context of an infection. Paul says, I have an appointment for my 5-year-old to get the vax next week. Wife is having cold feet now that she knows the size of the trial was 2,000, smaller than the adult trial. Why is this so?
It's hard to enroll children.
And Daniel Griffin addressed this last week. He got a question on his live stream, and he said, yes, it's small, but we also have the adult data, and we go
but
also by that.
Nonetheless, it's very hard to enroll children. It's like for any study that you do with kids, it's always much smaller because it's hard to enroll them, and then it's hard to do whatever the task is. I know this because we work on a childhood disease. And I've talked to a lot of pediatricians about getting certain samples and certain things, and it's very hard. And so the wife is having cold feet. Cold feet for what reason? So I think actually the best argument that you make is the one that is more analogous to why you vaccinate your kid against chickenpox, right? Because your kid is not going to be 5 forever. Kid's going to be 5 and then 10 and 15, and then lo and behold, your kid is going to be 40, right?
Yeah.
And so with that increase in age, the disease becomes more and more severe, right? So you're protecting, by immunizing your kid today, you're protecting them for the next 50 years of their lives, right? From a much more severe disease, which is why you basically vaccinate against chickenpox, because then there's no potential for you to get the severe form of chickenpox of reactivation, which is shingles, right?
Correct.
So I think actually if they had marketed it that way, it would have made a lot more sense to parents, and then we wouldn't have these stupid conversations.
Daniel Griffin mentioned on 86 that there's a 66% reduction in non-COVID death, like from stroke, heart attack, cognitive function. So this is an MMWR. And here's the question. Although deaths from after COVID vaccination have been reported to VAERS, the Vaccine Adverse Events Reporting System, few studies have been conducted to evaluate mortality not associated with COVID among vaccinated and unvaccinated groups. So they did that, and they found no increased risk for mortality among vaccine recipients. It reinforces the safety profile. So that's the conclusion of that study. All right, that's what it means.
Good.
Oh, you like my comforting home office. Thank you. Yes, the incubator is, I suppose, less comforting, but it will get more comforting as time goes on. It's a work in progress, for sure.
It's been in existence for what, a month and a half?
It's very short, isn't it?
I think so, but what do I know?
I think you know a lot. How do you feel about Noam Chomsky's comment suggesting that unvaccinated people should have the decency to remove themselves from society? What do you think about that, Amy?
How do you remove yourself from society?
Yeah, I just think that's not practical. But they have to work. They have to go to school. They have to do whatever it is they're doing. I don't think it's possible.
I think that they should practice physical distancing and wearing a mask if they choose not to be vaccinated for whatever reason. But
Right.
I don't know how you remove, and they should be aware of their surroundings and be very careful. Like if they do feel sick, that they should stay home and not push the envelope. But I don't really know how you remove people from society. What is that? You just build like a camp in the middle of the country and you put them all there?
I think it's unreasonable. Good. No, it's not a good idea. I think you have to deal with it in a civilized way, right?
Yeah, that doesn't sound good.
What would
but
be
I also don't know who Noam Chomsky is, do you?
Yeah. He's a very famous author, an American linguist, philosopher, cognitive scientist, historian, social critic, and political activist, the father of modern linguistics.
Okay.
Well, that's according to Wikipedia.
Okay.
Okay. Now, next question. What would be the recommendation for the interval between the second and the booster shots? Amy would say 6 months.
Well, the recommended interval is what, 6 months?
Yeah. Yep.
I would. Apparently you get shingles after the chickenpox vaccine. I did not know that. Did you? I thought that was the whole point.
I don't think it's 100% protection.
Well, nothing is 100%.
Yeah, you can get it, but I think it's much less in kids who have been vaccinated. So if you get a chickenpox vaccine as a kid, you don't get chickenpox. I don't know how you can get shingles. Right?
Right. But nothing is 100%, right?
No.
Except death.
Except death, yes. We're all going to die.
Right. So if what is the recommendation is 6 months between the second shot and the booster. I don't know what would be the recommendation. I don't know what that means. The recommendation right now is 6 months.
6 months. That's good. Yes, Miriam, I promised to send you a picture. I have your email. I'll get to it. Don't worry. I will send you a nice picture of SARS-CoV-2 virus. Does HSV 1 and 2 cross mutate? You mean recombine? Can we rephrase this question as recombination, Amy?
Sure.
And I don't know the answer. Do you? Do they recombine? I don't see why not. Is there enough homology?
I'm sure that there's enough homology, but the question is, do they, as far as I know, they infect completely different cells.
Right.
So how would you, is there ever a co-infected cell in your body?
Well, I think sometimes, so, herpes simplex 1 is oral, 2 is supposed to be genital, but sometimes Saul used to always say they mix up, they cross. So I think it could co-infect. Okay. And
well, then I guess it could, yeah. But I was under the impression that their tropism was mutually, it would be like polio and rhino recombining. Not sure that that's possible, as one is in the gut and in the brain and the other is in the lung. But Saul is the expert, so if that's what Saul said, I would go with what Saul said.
Okay. How do you get COVID from the blood? I guess you mean the deer blood. Well, you wouldn't, because it's not.
It's aerosolized when you chop it up.
From the respiratory tract, right?
Yeah, like when you harvest and you cut up, things get aerosolized that you didn't think should be aerosolized.
So were the deer PCR antibody positive?
I believe that they were both.
And so this high percentage of deer, they said they got it from hide samples. Wow. Deer skin. They found antibodies in the deer skin, which led them to this. The good news is it can't be passed from humans to humans from animals. How do you know that? Why would you say that, right?
Not clear to me that that's the correct. If that was the correct interpretation, then why did we call all the minks and stuff?
Right.
I'm not sure that that's correct.
If antibody aren't the correlative protection, it sounds like it's inaccurate to say natural infection is better than vaccination based on antibodies. It's reinfection rates. That's what we're using, right? We don't use antibody levels because you're right. We don't know what
antibody levels.
Absolutely.
And I'm not sure that natural infection isn't more protective or equally protective against reinfection as vaccination. I'm not sure that that will hold as more and more people.
How do you pronounce the different viral suffixes like viricota? Viricota. So it's viridi. Viricota is a higher one. Virales is another one. Okay, virales, viride, viricota. I don't remember what other ones are here. I should pull up a classification, but that's how you say them. The stated goal of the vaccines was immunity. What does immunity mean?
The stated goal, I thought, was protection against severe disease.
That's right. That's correct, not infection.
All right, 8 more minutes, and then it's 9 o'clock, and I need to put my overlay on and finish my ELISA.
Okay. If vaccines are meant to prevent disease and not infection, how do we stop the spread?
Well, you have enough hosts that have an immune response, and then it leads to abortive infections. And since it's an abortive infection, you don't have any infectious virus to spread.
That's exactly right. You both say the data not there to support boosters. Most of us are 6 months out from the second shot. Any thoughts on when we may need another vaccine? Are you waiting on more data?
Waiting on far more data.
I'd like far more data, but not to say that you can't choose to get a booster. If you ask me if I need it, that's what we will say based on the data. But you can get one for sure. In the UK, our governor is pushing the booster, but not advocating masks. Mixed signals. Shocking. Amy, your word has been adopted. Isn't that great?
It's fabulous. Everybody should use it.
Amy has certain words that she's made famous.
Yes, I have certain words that I've made famous.
Words and
not
phrases.
All the words that we should use. Yes.
With increasing numbers of at-home antigen tests being done, is the CDC ever aware of them? I doubt it, right? You do a test in your home and you tell your family or your workers or whatever. And no, I think they don't.
I don't know. I'm not sure how many people test positive and then go to the doctor.
They might. And then what happens?
Then the doctor records it, I guess.
Yes. But I think not everyone. You're not getting the negatives, that's for sure. We would like to know that, right? We're just getting the positives. And then we say, oh, we're still getting infections. Oh, my gosh. Shocking.
Yeah. Look, there's just lots of flaws in the system.
I didn't get my laptop yet. No, I'll let you know how it is. Are there university studies that confirm efficacy numbers from the pharma companies? Well, CDC, for example, continues to look at effectiveness. That's after the vaccine trial. It's called effectiveness. And they publish it regularly in MMWR. And there are likely university studies in progress, but we don't see those until they're published. Got my Pfizer boost yesterday, no side effects. Then I donated to Parasites Without Borders to offset any vaccine guilt. We need to vaccinate the world. So, folks, for the next 2 months, November and December, anything you donate to Parasites Without Borders, parasiteswithoutborders.com, they will match and donate to Microbe TV.
Which is great.
So it's a good way because they are a bona fide 501c3. And so your contribution is tax deductible, whereas the application for 501c3 status is mired at the IRS.
Mired, is that like the abyss?
It's like when you run your car into a mud bank.
Yes.
Michael says, I've seen claims that prior infection plus 1 vaccine dose is equal to or better than 2 doses without prior infection. Is there any evidence other than antibody level? So, Amy, I think it's mainly antibody levels and also the breadth of the antibodies, right?
Yes.
I don't think we have any real world effectiveness, right?
No.
Agreed? Yeah. Okay, Rima wants to know, what's the difference between Moderna and Pfizer? Gene sequence. What other vaccine components are different?
The lipid.
Yeah, the lipid composition is different.
I don't know that the gene sequence is different. There's only one way to encode for spike.
Yeah. What about the modified bases? Do you think they're different?
I thought that they were all pseudouridines. Maybe there's other differences. I don't know. I don't know enough about it. And the amount of RNA, but to be honest with you, it doesn't. Grams as a scientist doesn't mean anything to me, really. It has to do with moles. I don't know that is really a difference in moles.
Right. Victor says, do PhDs literally know every single detail of everything that's in the undergraduate textbooks in your field, like every single protein? Only Amy knows.
I don't even know that.
No, you don't. But you know where to look for it, Victor. That's the key.
Yeah.
Know where to look for it. So here is Priscilla. Amy, about Landers, I expected him to be like a minister of science, announce money, stuff like that. No, he doesn't announce money.
Money comes from money comes only from Congress. So he sits in the presidential cabinet. So I don't know how he would be in charge of money. He would have to be a member of Congress, right?
Yeah, Congress
I believe
allocates
that
money,
Congress
of course.
is the only entity that can define money.
Agreed. But the OSTP head just tries to promote science within the administration. And Joe Handelsman, a microbiologist from Wisconsin, used to be on Obama's OSTP, and she ran a lot of activities to educate kids about science. So they have activities like that. But Lander has a different role also in addition to that, because as Amy said, he's on the cabinet. One more, Amy?
Yeah.
NSP89 cross-react with NMDAR resulting in encephalitis. Can you break this down better?
I don't know anything about it.
So is this a case of self-antibodies against these proteins making antibodies that react with this receptor?
I don't know. I mean,
okay.
I don't. Isn't NSP8 and 9 part of the replication complex?
Yeah, I believe so.
So I don't know anything about it.
I suspect. Yeah, I haven't seen the paper, John, but it could be that they did this in cell culture, and maybe they're trying to make a correlate with. But encephalitis is not really a thing with COVID, right? It's more like other brain symptoms.
Yeah, it's not really. But the only, you can look at Niche and Ann Palmenberg's papers, and they'll tell you if there are antibodies against NSP 8 and 9 in people's serum, because they paneled all the protein products, right?
Yeah.
And then they tiled them by one amino acid. So, I mean, I'm not sure that there's antibodies against all the proteins, right? They'd have to be processed and stuff, and I'm not sure that all of them, are antigenic. I don't really know enough about it.
Okay, Amy, thank you for
All right.
joining
Off
us. I'll
to
talk to you
Eliza
tomorrow.
land. All right.
Eliza Land, I'll talk to you tomorrow.
Okay.
Bye.
Bye.
Okay, that's an explanation. Very good. Is it okay for a healthy teen to get 30 micrograms of Pfizer, wait 3 months, do 10? I don't know. I mean, if it's not the recommended dose, I mean, it seems to me that might be, right? So I just don't know what that would do. I'm sorry. Have you heard anything about Biden's invoking the Defense Production Act for producing at-home antigen tests? Yes, I heard that was a thing months ago. He announced that. But, yes, and they were to be distributed to people at either low or no cost. That would be great. I haven't heard anything. That would be nice. Can you elaborate on differences between infection versus vaccination in preventing future disease? All right, so Amy talked a bit about this tonight. The one thing we can't talk about future disease, right? There are some limited studies, but as Amy said, we'd like more. Observational studies are always hard because you don't always pick the population that you think might be appropriate for the study. But here's the thing. When you get infected and recover and you have one dose of vaccine, you make a high level of antibody, and the antibody is broader. It can inhibit infection with variants and also other coronaviruses. So it's a very broad response. So that's good, and it might be able to protect you against disease caused by future variants. That's what we know so far. Now, does that hold up in real world where people get infected? We don't know yet. That's the question that has to be addressed. You said it would not be a good strategy to get a COVID vaccine every 6 months, which could prevent infection. Why do you think so? Wouldn't it be quite a cheap intervention? I don't think it's good. I think you should make a vaccine that works and gives you long-term immunity. And we made a bunch of COVID vaccines that were good. They've saved lives. But apparently protection against infection. Well, we don't even want protection against infection. We want protection against severe disease. And so that's what the vaccines are doing right now. So why give a booster when we're still protected against severe disease? Do you want to prevent all COVID? Okay, if you don't want any kind of COVID, then maybe that's an approach. We don't know yet if that's going to work. But to do that every 6 months to prevent mainly common cold-like symptoms, I'm not sure that's good public health strategy. Now, I know we do flu vaccines every year, but look, in the U.S., we don't get half of the population vaccinated. People just don't like to get vaccinated every year for something. So that's in part why I think it's a poor public health strategy. I've heard that long COVID has been linked to EBV being reactivated. I would imagine CMV could happen too. Yes, I've heard about this. So these post-viral syndromes like long COVID and ME/CFS, they're post-viral. I don't. So ME/CFS, in many patients, they remember having a viral infection, which came right before it. And of course, long COVID comes after SARS-CoV-2 infection. It wouldn't be surprising to me that EBV or CMV would be reactivated during COVID because the infection is a stress. It's accompanied with inflammation, the production of cytokines, and that can trigger reactivation of these viruses. I'm not sure we know if they're involved in long COVID or not. I don't think we have any data on that, and I'm sure the studies are ongoing to sort that out. So we'll see. A Minister of Science could direct funding to basic research. So this lander doesn't have anything to do with funding. Discuss how research is made public. Yes. Discuss science with society. Yes. But he's not doing any of that. Now, what he should do, what Lander should do, is come on Twiv, right? But he would never do that. We're just small potatoes to him. And maybe you all should email him and say, you need to go on Twiv. I don't think he'll do it. Amy said last week she had a better chance of growing wings than a Pfizer booster creating new memory cells or strengthening existing ones. What is the science behind that hypothesis? Well, you'll have to ask Amy. And she's gone. But she doesn't think a booster is going to broaden the memory B cell breadth or an affinity and so forth. But you'll have to ask her why next time. I'm using the word story instead of narrative. Yeah, you guys told me you didn't like narrative. I thought it was a good word. It is a good word. But you have to change. You have to change it so you don't get boring, right? But I from time to time when I talk to new people, I do use it. Well, Daniel's Q&A, I understand that many people didn't know about it. I don't publicize at all. I really should. You know, I'm approaching 100,000 Twitter followers. I don't know why, because lately I haven't even used it, but I should have at least. I did publicize it on Twitter, but what else should I do? I don't think I even went to Facebook, where I have quite a few Twitter followers. But I do need a publicist. I'm trying to get one who would then, blast emails and whatever they do. I get emails from publicists every day. Hey, I have this great person you should interview. Have a look at this study. Unfolded protein response inhibition reduces MERS acute lung injury. So when stress induced by viruses can induce the unfolded protein response in the endoplasmic reticulum. And what they're saying here is that can lead to cell injury, which is not surprising. But I'll have a look at that. If someone has a rare reaction to a vaccine, does that imply that if they had gotten sick, they might have had the same symptom but with much greater severity? Daniel Griffin says no. There's no correlation or connection between how you respond to a vaccine and how you would fare if you were infected with, the agent that the vaccine is trying to protect you from getting infected with. Thank you, Ian. People who have questions don't always want them answered. That sounds like a wise saying, but I try and answer everyone's questions. That's what I'm here for, to answer your questions. Regarding the booster, did you or Amy once mentioned that an elevated level could be undesirable of antibodies? And so, yes, people have said this. If you do a boost every year or every 6 months, you're going to have high levels of antibodies. And so when you boost, your lymph nodes, your lymphoid organs make new antibodies. They continue to mutate the antibody genes and make antibodies that recognize different things. And if you continue to do this, the chance is you might make an antibody that recognizes your own tissues, right? And you would get an autoimmune disease. So that's one of the negatives of doing this. The same antigen over and over again. I'm not sure that's an issue because we get very similar influenza vaccines every year. But that's what they're talking about, yeah. Yeah, you always have to define better, for sure. I hope we do. But I always say if you want to look at effectiveness of vaccines, you have to define what you're looking to prevent. Don't just say effectiveness. Someone sent me a paper. Where is it from? From Sweden, which the headline is effectiveness of COVID vaccines in Sweden declines to, 60-some%. They're looking at all COVID. And if you look at the fine print, serious disease, not so much of a decline. You have to define your terms, yeah. I think the lack of viewers was because it was a one-off. Well, if there's a special on a TV channel, a one-off special, that doesn't keep people away. I just think as Someone just said, some people don't want their questions answered by an expert. And most people were quite civil on the live stream, actually, although we did have moderators doing their job. Thank you, Dan, or D. Wilson, for your contribution to the incubator. Elaborate on breadth of antibodies. So infection versus vaccine, different studies. Yeah, you're getting different info from different people because the studies are all different. Some studies say infection, greater breadth. Some studies say vaccine. I think it's highly variable. The one thing I can tell you is if you're infected, recover, 1 dose of vaccine, you make a really broad antibody response. You have the best immunity on the planet, most likely. Can a TWIV be done on the psychology and psychological effects of this pandemic and pandemics in general? Well, yes, we'd have to get experts on whom, I don't know. I usually get people in my field. I know them, and they say, yeah, sure, I'll come on to TWIV. Mostly, not always. So we have an author coming on Friday, the author of the COVID development of the COVID vaccine book, right, who happens to listen. And so he said, hey, I wrote this book. Maybe you want to read it. And he sent it to me. I read it, and I loved it. So I'm going to have him on to talk about it. But yes, we need help in identifying the right people to do that, principal manager. But I would do it, yes. Yes, you were right that most people will have watched the Daniel Griffin thing after the fact. So I can tell you right now that the Daniel Griffin, where is it? It's already flowed off. There are only 5 recent videos on the top, and it's already flowed off. Yeah, it has 12,000 views, the Q&A. That's pretty good. His Q&As, well, they're not Q&As, they're clinical updates, right? They get 11 to 12,000 views typically. So but I thought it would be much greater. There are other people on YouTube who get millions of viewers, and they're far less authoritative than Daniel. That's my point. And you're right, it's better than nothing, so I'll stop complaining. I just wanted to tell you I was a little surprised, but no big deal. No big deal. Did you see a paper called Membrane Fusion and Immune Evasion by the Spike of Delta? Suggests it might account for increased transmissibility. So I have seen this, and indeed some of the changes in Delta may likely contribute to evasion of antibodies for sure, but whether that contributes to greater infection, we have no idea. And these papers all use transmissibility very loosely and incorrectly, in my opinion. The Delta variant is more fit. It spreads very well in populations because it displaces other variants. Whether it's actually infecting more people on average, we just don't know. Although the epidemiologists will say, oh, sure, we know. They don't. We need more work to do that. So unfortunately, many papers just repeat that word, transmissibility. It gets repeated over and over, and people think that's it, but it's not. Market yourselves. Brian, I can't do it myself. I already pretty much have overextended. I can't even do the work at the incubator that I need to do to get it in shape, to test equipment and so forth. I'm not complaining, but I need a publicist. I can't do it myself. I'm 80. I did get Moderna. I got a booster. That's great. You have no idea how protected you are. You're right. You probably don't need to stay home. If you want to do something, you could go out and wear a mask. I think that would be fine. If you've got 3 shots now, at your age, you should be okay. Wouldn't a booster further impede spread? So here's the story, Taylor. It would. Right now, spread is already impeded by population immunity. It's not going to be shut off. It's not like a switch where we get to a certain percent and there's 0 transmission. It's just reduced substantially. If you have high antibody levels in your blood shortly after immunization, then yeah, you won't even be infected. But those antibody levels decline. They always do. That's the normal physiological process. Antibody levels don't stay high. And so within 6 to 8 months, you can be infected, and it takes 2 or 3 days for your memory B cells to start making antibodies, those antiviral antibodies. And in that time, you're infected. The virus has entered your cells. It's beginning to reproduce, but then the antibody is made by those memory cells and it clamps down on it. In the meantime, you could be shedding, and it depends on the person. Some people will shed barely anything, so they won't transmit. So my point is that I think the vaccines do a good job of muting transmission. Sure, every time you had a boost, you'd probably not be infected for 6 months, but then you'd need another boost. And I don't think that's good public health policy, frankly. For reasons that I discussed above. Could long COVID lead to Alzheimer's? Could be. The protein clumping that we see in the brain, the amyloid deposits, that seems to be a response to virus infection. And so it could, but I think the invasion of the brain by SARS-CoV-2 is minimal, so possibly not. Is Delta Plus more dangerous? Well, I think we'd needed a year before we'd know that. There are going to be a number of observational studies that claim it is, right? Just as they claim Delta was more dangerous than the previous. But then with time, as you do the studies more carefully, you see, no, there's no difference. Yes, we're going to put decorations in. This is one of the things I haven't had a chance to do. I have to move the desk. I have to put a shelf together that I bought. I want a shelf behind me to put models of viruses. Here, let me show you one. I don't like to pick these up. Amy doesn't like me to pick them up during the show. So this is a coronavirus model, right? Isn't it gorgeous? Put it next to me so it'll be in focus. Those are the spikes, see? And the membrane is in gray, and then it's cut in half so you can see the RNA. I want to put these on a shelf behind me. And I also have a nice picture, a watercolor of viruses that I commissioned from Michelle Banks. I'm going to hang that up. But, I get to the incubator and I start recording, and then it's time to go. So I'll get to it. I will. I take it. I understand that there's not enough decoration, especially compared to this, right? But, I've been here since 1989. It took a long time to get all this stuff here, right? By the way, that's a watercolor by Michelle Banks. Really nice. The viruses. I'm 62. I got the MMR in November. As it was said, many longer had protection 50 years later. Plus, I had a newborn grandchild. I got COVID 3 years later, didn't protect 3 months later. Yeah, Marty, but you know how it goes. It doesn't work in everybody. It's not 100%. So we can't take away much from your experience, sadly. Got a COVID antibody test. My level was 0.62 units per mil. The cutoff for a positive was 0.8. I was seriously ill in January 2020, but was not tested. I don't know what this cutoff means, but I don't know if that has anything to do with you being infected. I think it's an arbitrary cutoff. Well, Amy's gone. Do you believe that a single infection is better protection against disease than 2 doses of mRNA or a single dose? I'm not going to answer for her. I'll let her do that next week. Yeah, sitting next to each other gives a dynamic. I agree. I think it's very different when we're on Zoom, on the separate places, but unfortunately, that's the way it has to be because I can't be in there every week. A significant number of those who claim long COVID did not have COVID. Is long COVID a real thing? I think yes, it is a real thing. It's quite confirmed. It has a diagnosis now, the numbers. It's a real thing. It's a syndrome. We know all the different aspects of it. Yes, there's no question about it. A cruel world. Do masks stop a virus as small as COVID? Oh, yes. It's not stopping the virus, actually. It's stopping the respiratory droplets that you're exhaling and coughing out and sneezing. Stopping the water, the droplets, which are much bigger than the virus. Yeah, the virus would go right through a mask, but we don't exhale just viruses. We exhale viruses in droplets, and those get caught up. I'm really interested in viral DNA in the human genome, like the placenta example. Where can I read more about this? Well, there's a lot in the literature. You've probably heard me talk about it at some point, maybe in the course, I don't know, Virology Live. But there are whole articles about it. If you want me, email me, Vincent at microbe.tv, and I'll send you some articles if you don't want to search for them yourself. Thank you for your contribution to the incubator. It's appreciated. Now, a lot of discussion about masking. I'm moving through these questions tonight. It's good. We have 577 people. I do hope some of you are staying for the long term. The questions for Amy I'm going to skip because she's not here and I shouldn't answer for her, right? You just donated $10. I don't know where it went. If you donated $10, it should come up in the live stream here. If I don't see it, it probably didn't work. And you know, Amy predicting of the pandemic going 2 more years, you have to ask her to explain it. Okay? I don't want to explain it for her. Do you think experimental medicine will follow up on the cinnamon and hop extracts as potential immunomodulators? So, you know, many drugs are natural products. They start as a natural product. People screen cinnamon and hop for inhibitors, and they find them, and then they purify the inhibitor, and then in many cases they can synthesize it. So it could be. It depends how specific this inhibitor is. If it could hit other targets, and that's not good, and how potent it is. So it's hard to predict. Most hits fall by the wayside and never get pursued or never turn up positive. Do you agree with Condit that eventually everyone will either be vaccinated or infected? I do think that's correct, yeah. This is it's just moving through the population. It's not going to happen well now. It will happen in the coming years, just as most people are infected with many other viruses like rhinoviruses. And Amy has arguments for 5 years, the 2 more years. So you'd have to talk to her about it. She will explain it very well. Pfizer going to tris buffer for kids version instead of phosphate. So buffers maintain the pH of a solution, right? And there are many ways that you can buffer the pH, that is, make it resistant to pH change. And phosphate-based buffers are relatively weak. Tris-based buffers, however, are much stronger, and so they may have found that gives it better stability. You know, as they're testing, they find this out. So that's not surprising. Comment on this Armed Forces personnel saying the vaccine showed only 3% effectiveness. 3% is just ridiculous. I mean, the placebo effect is 30%. So there's something wrong there. And I haven't seen this study. So we will comment on it when it comes out for sure. Malawi. Wow, I'm impressed. How are the fish? How are the cichlids? I used to have cichlids. Do you think places where cases are low and vaccination is still in the 50, 60 will inevitably have more waves? Probably. The virus will find its way everywhere with travel, especially as the holidays are coming up, right? You could get a booster for a holiday. As I always say, look, you can have a booster, but if you ask me, do I need it? I'll tell you why I don't think you do. That's all I'm about here. I'm just an honest guy. I don't have any horse in this race, as they say. Chickenpox, herpes, shingles are these three different stages of the same virus. So one virus causes chickenpox and shingles. You get chickenpox as a kid, you get a rash. The virus goes latent in your nerves, which means it hides there. And then typically in your 50s and older, it comes out again, it reproduces and gives you a rash on your skin. So that's called shingles compared to chickenpox. Herpes is a different virus. It's in the same family, herpes simplex viruses. Same family, herpes simplex viruses, chickenpox, shingles, or the virus is called varicella zoster virus, VZV. Then there's Epstein-Barr virus, cytomegalovirus, and others. They're all herpes viruses. So they're not the same virus. But chickenpox and shingles is the same virus, yeah. Is there a list of your appearances on other channel YouTubes and pods? You want me to make a list? I can do it. Make a list. I'm not going to do it beyond this year, probably. I think I have list of appearances. I don't know when I'm going to get to that, but I'll try and do it. You do get shingles after the chickenpox vaccine. So, yeah, I mean, someone said earlier chickenpox vaccine isn't that old, but that may be right. I don't remember offhand. Shingles after chickenpox. Let's see what the frequency is. It may be not so frequent, right? If you've been vaccinated with the chickenpox vaccine, it's possible to develop shingles since it is an infectious vaccine. But I have to see that the issue is we didn't know for many years because it takes a while to get shingles after the initial infection. So I think that is quite rare, most likely. Why does the second dose have more side effects? Well, your body has seen the vaccine already, and it remembers it. You have memory of it in the form of B cells and T cells that remember, and then as they see it, they respond really quickly and forcefully, more so than the first time, and that's why you may get a stronger response, but you don't always. You don't always. Well, do you think other SSRIs will work in helping those with COVID? Daniel is not sure. He said, don't take other SSRIs until we test them. So I just don't know. Yeah, so Patricia is in response to my a statement a while ago, which was that, don't take people out of society. That's not right. And she said that's idealistic because people who refuse also don't mask or distance. Yes, I agree. And that's the philosophy of that sentiment, right? Because they're not separating themselves when they should. So they're being rather selfish. I agree. I don't know what to do about it. I think that's a psychological, sociological decision. I ask people why they don't want to take this vaccine. And they say, well, I'm afraid of long-term side effects. And you know what I tell them? Look, we don't know of any for any vaccine. And sure, I can't tell you that they're going to be zero for this one, but probably not. But anyway, why don't you just be brave and take a chance? What is it? You're worried about a side effect when you're 90? The first day I was able to take the vaccine, I did. And many other people did. I don't get this. I don't think it's that. I think that they just don't want to be told what to do. Well, then fine. I agree. It is a conundrum. Given over 40 million reported cases in the U.S., do you agree that almost 4 times or 160 million have natural immunity? If accurate, do they need to be vaccinated? I don't know if 4 times. I think that it's been underreported at least 5 times, right? But how many have good immunity, we don't know. That's the problem. We just don't know. And so we want to make sure they're protected by at least giving them 1 dose of vaccine. I don't think that, the varicella is old enough for people, but I think now we're starting to see some shingles, as I just mentioned before. There is a new and improved shingles vaccine, right? There's the old one, which was really a higher dose of the chickenpox vaccine, and then that was also an attenuated vaccine. So the new one now that you should get for a shingles vaccine is called Shingrix, and it is a viral glycoprotein from the virus produced in insect cells mixed with adjuvant. It's very good. It has a very high effectiveness. Amy is truthful, straightforward. She tells you what she's thinking. You may not like what she says, but that's what she feels, and she doesn't obfuscate or lie or prevaricate in any way. She's really, what you see is what you get, folks. And why don't other people do that? I don't know. I can't talk for others. This is funny. Ricotta cheese or ricotta cheese? Yeah. You roll your eyes at the unvaccinated, but natural immunity is science. It is. But if we say you should be vaccinated to protect others, why can't you just do that? Why? And that's what she's rolling her eyes at, okay? You need to understand that a cruel world. Hey, that's good. A cruel world, you bet. Do viruses infect bacteria? Yes, they do. If yes, does this affect human cells? So you have a microbiome in you, right? You have bacteria in every one of your different organs and tissues. And that microbiome, its composition is influenced by the viruses that infect it. So yes, the viruses of bacteria can affect us, although they can't infect us. Why is it a good idea to vaccinate kids with a vaccine that's shown to have a higher risk? It's not a higher risk. COVID has a far more higher risk of myocarditis than the vaccine. And the myocarditis associated with either is treatable. Actually, the vaccine-associated myocarditis is very, very treatable and typically transient. So, Elle, you got your facts wrong. I'm sorry. What is the common definition of severe disease for which the vaccines provide added protection? That's a great question, S-Son, and it varies according to the study. You know, the one that we can agree on is death. We know what death is, right? Hospitalization is even squishy because in different places you may be hospitalized more or less quickly, and certainly depending on how old you are. So that's squishy. And then severe disease, yeah, what does that mean? You have to give some metrics to it. Is it you're in the ICU, you're on high flow oxygen? That would be good with me, but that's not what it is in all studies. So this is a problem to a certain extent, that there's variability in the reporting of what is or the definition of what is exactly serious disease. All right, we have 584 people here. Could we get the likes over 300? Actually, 400 would be great. All you have to do is go below the video window and push the thumbs up button. We'd appreciate it. I don't get money for it. It's not the point. It helps to spread the word of science. Does anyone know for March 7, 16 where affordable rapid tests can be found? 10, 20, 70 in Virginia. Anyone knows to help her out, that would be great. Are monoclonals going to replace chemo? I don't think so. I think they're going to be part of a multi-pronged strategy, which includes, checkpoint inhibitors and CAR T-cell, NK cell, MAC cell therapies. I think it's going to be not just one thing. But I'm not a cancer guy, so I'm not the expert. We don't go around saying people have tested positive for AIDS. We see HIV for a reason. Asymptomatic cases should not be called COVID. Oh, this is a very good question. So what is a symptom? That's the question here, right? A symptom is something you feel. But you may not feel something, but things are happening. You have signs of infection. You have the virus. The genomes are replicating. You can detect that. You're making an immune response. You're making cytokines and so forth. So you're making signs, but you don't feel those, so they're not symptoms. So you say, the doctor says to you, do you have any symptoms? And you say, no, I'm symptom-free. That's what asymptomatic means. But you're still infected, and you have a virus reproducing you, so you have COVID, even though you're asymptomatic. I think COVID is the right way because coronavirus infectious disease, right? That's the whole spectrum from asymptomatic to death. But that's just me. You don't have to listen to me. I'm just here 2 or 3 times a week. Liking is never having to say you're sorry. You could like and say you're sorry. Now we're up to 343. Thanks, folks. You're really nice. How much time infected person can be infectious for other if severely ill? So the total infectivity is no more than 10 to 12 days. And if you're severely ill, you're way beyond the point where you're infectious to others. I think the greatest time of infectivity that's been reported is like 21 days. But most of the time, if you get severely ill, your virus titers are already low, and I don't think you're transmitting to others. As we get more data on long COVID, do you think the burden of disease, especially long COVID, will affect the population structure? I do. I think long COVID is going to be huge. It's going to be a huge national tragedy, international tragedy. Someone said earlier there are other countries. I know. I understand. I understand that most undeveloped countries are less than 5% vaccinated. If I could personally do something about that, I would. I'd go vaccinate people, but they don't need me to do that. We need vaccine. So I do think this is going to be a problem. It's going to put a burden on healthcare, and I think we are just seeing the beginning of it, in fact. Thank you, Patricia, for your contribution. Really appreciate it. Studies show that there's more viral load in the obese. It's because obesity is an inflammatory disease. Could this be why healthy people are asymptomatic? I don't think so. I think why people are asymptomatic is unknown. They're reproducing virus, but no symptoms, which means they don't feel anything, but the virus is still doing things in them. Is there any chance of getting the next pandemic from pets? That is an interesting question. And in fact, it's been suggested that dogs in China might be a source of the next pandemic. Now, I don't mean to start any bad rumors, but apparently dogs have become very popular in China in the last, 10 to 15 years. So before that, they weren't so popular. Now, people are crazy about dogs in China. So a lot of people have dogs. And as you know, there are a lot of people in China. So there are a lot of dogs in China. And dogs get influenza virus, and they have influenza virus there. So they're thinking, so far we've seen new pandemic influenza strains come from pigs and birds. It could come from dogs. So, yeah, it could be. Ohio's distributing free C19 home test kits. That's great. Wonderful. Every state is different. I don't mind getting vaccinated once a year. John, bless you. Not everybody's like that. Witness the low uptake of influenza vaccine every year. Someone has cold symptoms 3 weeks later, sudden fatal PNA, rapid test at 3 weeks negative. it might not have been SARS-CoV-2, right? Could be something else, but not every infection is positive as either. And Daniel Griffin talked about that last week. So I'm not surprised, no. How do we fight COVID misinformation? Encourage vaccine hesitant to get vaccinated? Well, tough questions. You have to identify sources of good COVID information, like this live stream, my podcast, and others who do similar good things. There are a few other channels on YouTube that have good information. And, if you can talk to someone who's vaccine hesitant, ask them why. What's your worry? And try and talk to them about it. As I've said, if I could talk to people one-on-one, I'd love to understand their hesitations and try and explain. And, a lot of it is misinformation they're getting, and you can fix it. Unfortunately, a lot of people who are vaccine hesitant get angry and start yelling and screaming, and then the conversation degrades, and it's not productive. But I'd be willing to talk to people anytime. We need more science communicators. I don't know about me. I do my best, and Amy's a great sidekick. I don't want to be a minister of science. I like to do this. I like building out the incubator and doing all that stuff. I'm not good at managing large numbers of people, I think. Yeah, so this virus hunter's, his wife, Christine, who directs these virus hunters in collaboration with him, sent me this preprint. And maybe I'll have her on to talk about it. So she's a PhD. She works for a company, a biotech company, and directs these New York City virus hunters to do this. It's a very interesting project. Yeah. It would be great if your website had a calendar of appearances and guests. Yes, I agree. What seems to have been forgotten about boosters is that they were originally recommended for over 65 immunocompromised. Yes, exactly. That's exactly right. And I have no problem with that, even though I think the data saying that they need it is there. It's fine. But now it's gone to everyone. In fact, yeah, some states I've seen in the chat here. Anybody, is it Australia? Anybody can get a booster. Yeah, Mr. Ozzycam said that. It's not the original purpose. You can use tail in place of story. And yeah, there are many words you can use. I like to play with words. Does that principle not apply with the yearly vaccine due to being a different strain? You mean the idea that you're going to get autoimmunity? Well, that's what I said. I don't understand. It should be. They're very similar. There's a lot of conserved antibodies in those viruses, and then those B cells are maturing and changing. So I don't see why autoimmunity wouldn't be an issue with that as well. UK is vaccinated around 70%, but cases are still trending up. Is there a known vaccination level populations need to hit to turn the tide long term? Well, this is another squishy issue, this herd immunity number. So it's based on the reproductive index, which can only be determined accurately early in the infection. And even then, it's not fixed. It can be influenced by human behavior. So it's not just a virus property. So for SARS-CoV-2, it was 2 to 3. So that means 50 to 70% of the population needs to be immune to impact transmission. So obviously 50% is not working. Maybe it's because of immune evasion of the variants, as it is for influenza viruses. But I think 80%, 70% vaccination, you must have 20 to 30% infection and recovered, right? So you should be at herd immunity. But if you have, herd immunity is nullified if you have pockets of susceptible people that are together. And there the virus introduction spreads through them because none of them are immune, so there's no herd immunity in them. The rest of the population has herd immunity, which means reduction of transmission, but not them. So that's an important caveat to remember about herd immunity. Smallpox has been eradicated, polio virtually eradicated, both due to vaccination. Correct. This will never happen with SARS-CoV-2 because it has a property that the other two viruses do not have, and that is SARS-CoV-2 infects animals other than people. So there's always a reservoir of virus around that could infect us. Smallpox only infects people. Poliovirus only infects people, so they can both be eradicated. And that's the simple answer. Why is tinnitus much worse after the vaccine shots? No idea. I don't know. Worse than what? As compared to infection? I mean, I think in both cases you're having inflammation that's generating the tinnitus. Do you think it's a good idea to do a risk assessment comparing risk of serious illness from SARS-CoV-2 versus the same from vaccines? Yes, for sure, and that has been done. And for myocarditis, it's far, far higher from COVID than from vaccines, for sure. There's no question. And that's why, even though certain vaccines are associated with myocarditis, it's still recommended to take them because their benefit outweighs the risk. Everything in life is a risk-benefit calculation, whether you know it or not. You know, when you get in your car and drive to work, it's a risk-benefit calculation because there's a risk of getting killed. There's a risk of harm at everything you do in the world, and vaccines are no different. That's something that kind of is weird, right? People want vaccines to be 100% safe. They're not. There never will be, but neither is life. Did I hear on Twiv last year that some people's reaction to the vaccine could be due to or made worse by the fat that is wrapped around the mRNA, which is slightly different than natural fat? I think that yes, so your response to the lipid nanoparticle could in part drive inflammation. The RNA itself would not do that because this was the magic about the mRNA vaccines. It was found that if you use modified bases, not normal mRNA bases, but chemically modified bases, it reduces the innate immune recognition of the mRNA. And so what's left is the lipid. But I'm not sure that that's a fact, that's a speculation. I'm not sure that that's been shown experimentally. Yes, and Twitter is not a good place to reach followers. You know, there's no one good place to reach everyone, right? Only if you're president of the U.S. and you get on TV, then you can reach, 200 million people, but not everybody. But I have to, that's why I need a publicist. They'll do it, right? So this is funny, the time of day, 5 p.m. Yeah, but when I do it at 8, people say it's too late. If I do it at 11 a.m., it's too early. You can't please everyone, right? You just have to do it. You just have to do it and get your best, right? But the recording's there. Yeah, shot to save the world, right? Gregory Zuckerman, and he is on Twiv Friday. I hope you enjoy it. On vaccination against variants versus previous infection, I think Paul Benash also said that people in the vax group fall more consistently in the high antibody group, although the average is similar. That's right. And depending on the study you quote, you're going to get a different outcome, yeah. Stephen Taylor wrote a book called The Psychology of Pandemics. I would like to get someone like that on, sure. Nicholas Christakis for the social aspect. Okay, good. We have 2 suggestions there. Ooh, a web app called Eterna. It's a puzzle game based on RNA design. Ooh, that would be fun. Maybe Conda and I could play it on a pod sometime. Or Brianne or Kathy or... I don't know about Dixon. Alan maybe. Alan's a gamer, right? Do you think we will ever be able to engineer protists to target specific viruses and not cause harm to human health hosts? I don't know why you'd want to do a protist to do that. And I'm not sure you want to give people protists, right? So I don't think so. No. So that was SARS-CoV-2. Can I show you my other model? Oh, this is gorgeous. This is poliovirus. It even has a plaque. Look, poliovirus plus PVR, poliovirus receptor. This is not something you see in nature because it's artificial. It's the receptor, purified receptor bound to the virus, and then the structure was determined. But I had this done. I had it made. You know, it matches the icon you see on all the TWIVs. The colors match, magenta and blue. Isn't that gorgeous? I love it. This is going to go in the incubator on the shelf behind me. So those of you, it matches my glasses, right? Those of you who are complaining that the wall is bland, it'll get better. Chill. Just patience, relax. Grad student in physics. I'll work for you for free. I don't take people for free. I don't think it works. I think you have to pay people. Now, the moderators aren't getting paid. I'd like to. I just feel that these moderators are actually spectacular because they do their job without being paid. But I've had many volunteers over the years. They volunteer for a little bit and then they're gone. So I find if you pay people, they stay. Okay? So mods are different. But any of you mods, if you decide to leave, don't. Just talk to me first. How is oxygen in cells affecting the virus? Well, oxygen, of course, is a terminal electron acceptor in the respiratory chain, and we need it to make energy. So more oxygen, more energy, more virus reproduction. So more oxygen is not going to decrease the chance of severe COVID. Now, if you can't breathe because of the lung problems caused by COVID, then you need to have oxygen pump into you. Otherwise, you die because your tissues won't be able to make enough energy. Is the antibody tested in micrograms per mil? No, it's in international units, which are calibrated to be a certain amount of neutralization. Thank you, Robert, for your contribution to the incubator. Really appreciate it. I learned in relation to cancer, T cells require certain free fatty acids and are in competition with cancer for these nutrients. Is free fatty acids in play? Well, yes, viruses require free fatty acids for their membranes. Not just the virus membrane, but also in the cell. They make membranes to reproduce their RNA on or in. And so I don't know that we know for SARS-CoV-2, but certainly I'm sure it will turn out to be that probably infection induces fatty acid synthesis and may even inhibit oxidation of fatty acids, which is a process that will generate energy, but the virus doesn't want that to happen. Or viruses don't want anything, but it's not beneficial for reproduction, so it's inhibited. How is this mRNA created? So they start with a plasmid, which is a piece of double-stranded DNA. Actually, it's just a linear piece of DNA that can be synthesized. And then they have a sequence that's recognized by an enzyme, RNA polymerase, that will then generate lots and lots of RNA in a test tube. And they make kilograms of mRNA. It's amazing. It's amazing. You want me to put this? It's not a Strat. It's a poor man's Starcaster. Okay, it's only $200. I'm not good enough to warrant a Strat. And I bought it years ago when I was a young guy, and I don't play it anymore because I'm talking to you guys and thinking about viruses most of the time. Let's see. I've been drinking beer with hops, knew it was good for me. There's something in everything, but the concentrations are too low. All right. Some in the U.S. say the virus is coming to its limit of variation. Nonsense. Viruses always vary. There's no limit of variation. There may be limits as to what the population selects out of the variants, but there's always variation and always an opportunity for a new variant to arise. So they're nonsense. They're nonsense, and they're just being influenced by, falling rates in many places. What, if any, impact does the discovery of circulating exosomes after infection have the program of subunit vaccines? I don't think it has any impact on subunit vaccines at all. It's a separate issue and not related, really. Do we know that our DRP primers detect SARS-CoV-2? Yes, we can do that. They're typically made to other parts of the genome, but you could do any part of the genome. Could the virus mutate to a level where the vaccine has no efficacy? No. Nope. Not happening, because the T cell epitopes don't change in these variants. Only the B cells, and the T cells would save your life. Is there going to be one more COVID wave? Somewhere in the world there will be, for sure. Amy thinks for the next 2 years. Yep. SARS-2 was found in olfaction. Was that an infected cell or particles? So it's in infected cells. They're the sustentacular cells, not the neurons in the olfactory mucosa. It's just one synapse away from the limbic system. I think the evidence that the virus gets into the brain is very low. I think rather the chemokines and the hypoxia are what are responsible for the majority of neurological symptoms. There are already a lot of people taking SSRIs, I know. But Daniel said there's no evidence that they're going to help with COVID. Maybe people will do observational studies that look at that. That would be interesting, right? This is cool. If I get a spike growing out of my head, I'll put a hat on it. Be brave, folks. But the folks that are afraid are not here, right? Yeah, the new chickenpox vaccine is just the spike, one of the glycoproteins of the virus, yeah. People shouldn't worry about the vaccine, really. I think it's other issues that are inhibiting them, and I'd like to know what it is. If it's that I don't want someone to tell me what to do, okay, then decide on your own. How about for that? Nucleocapsid is the most abundant protein in the virus. Why do you think symptomatic post-vax won't make antibodies to it? So Daniel has said, and this came up in the paper we did last week, that Vaccinate, you could challenge a vaccinated non-human primate, for example, and they test PCR positive, and they have virus in them, but they don't make antibodies to N. So they're not probably infected, actually. So that's my take. And Daniel agrees with that. He's seeing it in people. I took the vaccine. I don't get why we have to mask. Well, the masking is because some people, the CDC feels that transmission is not completely inhibited by vaccination. I think the data are not so good. But, in many places you can't go without a mask, so fine. But if you left it to me, things would be different. If lipid nanoparticle carrying the mRNA ends up in parts of the body other than the shoulder, could this cause any mild or moderate issues? We don't know. I think not, but some people think it may be involved in myocarditis, but we just have no evidence to indicate that. Is there going to be a major resistant bacteria emerging from the hygiene theater? I don't think so, not necessarily. I do think our immunity, general immunity, is compromised somewhat, but I don't think it's going to lead to that. I think antibiotic overuse is going to be a real issue. It already is. Crotty may disagree with Amy. Yes, I'm sure she disagrees with Crotty. She can tell you about that. During the Australian bushfires, all states that were severely affected were also that had high COVID. Is this bad luck or smoke related? I don't know. Did people inhale enough smoke to make a difference? We do know that smoke can compromise your respiratory defenses, but that's, chronic smoking, and I'm not sure how chronic this was. So it might have just been bad luck, and unfortunately, we will not know, likely. Your bone marrow transplants are contagious. How do you know they're contagious? Don't tell me they're PCR positive, because that doesn't tell you they're contagious. But certainly immunosuppressed people could shed for longer periods. I just questioned contagious, how you know that. Have you already had an in-depth conversation between humans, microbiome, and viruses? I don't remember. Probably not. But I'm sure it'll come up at some point. Absolutely. How can we explain asymptomatic cases? By cross-reactive T and B cells to epitopes conserved, in addition to lower allostatic load in healthy people? I think it's a lot of things. I don't think it's one thing. I think part of it is probably a very good innate immune response initially in infection. There might be some contribution of cross-reactivity, but who knows? Nobody has studied that. There may be a genetic predisposition as well. You have certain immune genes that are really good compared to others. Just don't know. What do you make of the Israeli data that shows higher estimated efficacy for documented infection in pre-infected persons with natural immunity than in persons who are vaccinated? Well, that's about the same, I would say. So that would support the idea that the protection is the same in either case, right? But I would say that you have to take those data with the limitations that they're an observational experiment without really good controls, right? Thank you for your contribution, Margot. Really appreciate it. Supporting the incubator. Coxsackie virus does cause pericarditis. Others do as well. All right, let's, it's already past 10 p.m., so let's quickly scroll through. I think your Jillian Michaels type conversation will do more for the vaccine hesitant than on the shaming. Yeah, so I have had a change in tone. Certainly early in the pandemic, I used the shame approach, but as I started to get on shows like Lex Friedman and Jillian Michaels and others, I think a more understanding approach is beneficial. Like, here's what I think and why, what's your issue, and talk to me about it. I think it works better. Is it possible to have a COVID reinfection while testing negative but still have symptoms? Sure. But, if you have an antigen test and it's negative, you should have a PCR test just to make sure, right? Thank you, Kathleen, for your contribution. Really appreciate it. Why do we need 5 to 11-year-olds to be vaccinated? Because if that 0.005% is your kid, you don't want your kid dying, do you? So you're not thinking lazy hook about someone else's kid. You're just thinking about your own. Thank you, Slowomir, for your contribution. Really appreciate it. Yes, and this is right, Typhorons. I think mRNA vaccines use a mix, different bases as an adjuvant. Not different bases, but they're modified bases. The uracil is modified chemically, so it's ignored by the innate immune system. You don't get a bad reaction to it. I need a publicist and a feng shui consultant. Okay, so you don't like my feng shui. It's fine. It doesn't bother me at all. Other things would bother me, but not feng shui. Is the spot on the wall being painted? No, I put a picture over it for now, but eventually there'll be a big picture of it. Over it. I've been making winter scarves. Good. Today was cold here in the Northeast. Oh my gosh. And I appreciate all your thanks on top of it all, for sure. And I know I won't get to all of your questions, but do come back next week. Thank you, Faster Tortoise, for your contribution. Really appreciate it. Let's see here. Yeah, don't live your life worrying about tiny risks. Okay, folks, that does it for another Q&A with A&V. 600 people for 397 likes. Push it over 400 in the next 30 seconds while I say goodbye and thank you. Thank you to all the moderators. You know who you are. You know who they are. And thanks all of you for coming. I appreciate your questions. Quite civil tonight for the most part. Not aggressive. I don't mind being questioned, but nastiness isn't needed. So come back next week, Wednesday. Next Wednesday, I'll be back with Amy. Q&A with A&V. In the meantime, please be safe, everybody. See you next week.